Today we will start a new chapter, chapter 5. The name of chapter, membrane targeting of proteins. Different types of protein, membrane protein, carrier protein, and different types of protein. How can it target in different cellular organ? First, membrane protein. What is membrane protein? Membrane protein, what is membrane protein and what is the roles of membrane protein? Membrane proteins perform a variety of functions vital to the survival of microorganisms. Membrane protein relay signals between the cell's internal and external environment. Transport proteins moves and ions across the membrane. Membrane proteins expose surfaces that are readily suited for incorporate into or binding to membranes. So you see, membrane proteins some specific function and play some vital roles in the cell to the survival of organism. To the survival of organism and relay signal between the cell's internal and external environment. Internal and external environment of the cell inside transfer different types of signal, and also incorporation into or binding to membranes. Examples of integral membrane protein and their functions are some integral membrane protein. Membrane protein are different types. Integral membrane protein, first example, glycoprotein. The function of glycoprotein, cell to cell interaction. Another protein, sodium potassium, sodium ion, potassium ion, ATP, ATPase, responsible for establishing and maintaining the electrochemical gradients of sodium and potassium ion across the plasma membrane. Glucose permease. Glucose permease, another integral membrane protein. The major function of the glucose permease, the reversible transporter protein of glucose. So, membrane protein. You read for your April 2021 exam. First you read what is membrane protein and examples of membrane protein with their function. It is important, and I know it is a new term and newly assigned term in your new sheet. Types of membrane protein. Membrane protein are different types. First, receptor protein, peripheral or intrinsic protein, integral, extrinsic protein, integral, intrinsic protein, transport protein, adhesion protein, glycoprotein, channel protein, carrier protein, signal transduction protein, recognition protein. Those are the types of membrane protein. Function of membrane protein. Already we know some specific function of membrane protein in the definition part to the survival of microorganism. The vital function of the membrane protein to the survival of microorganism. Another function, relays signals between cell internal and external environment, and another important function, expose surfaces that are ideally suited for incorporation into or binding to membranes. These three specific function you may include in the function area. Another some function, membrane protein mediate processes that are functional for the furnishing of biological cell. Membrane embedded transporters move ions and larger solids across membranes. Receptor mediated communication between the cells and its environment, and membrane embedded enzymes catalyzed chemical reaction. Attachment to another function, attachment to skeleton and cellular matrix, maintains cell shape, stabilizes skeleton. And some other important functions, intercellular joining, enzymatic activity, cell to cell recognition, transportation, signal transduction, cell recognition, intercellular joining, attachment. Those are the another function of membrane protein. So membrane protein must be read in your April 2021 exam. It may be short note question or definition, an example with function times or function of membrane protein. Next term of your sheet, secretory pathway. What is secretory pathway? The secretory pathway refers to the endoplasmic reticulum, Golgi apparatus, and the vesicles that travel in between them, as well as the cell membrane and lysosomes. Secretory pathway generally involves some organelles, endoplasmic reticulum, Golgi apparatus, and vesicles that travels in between them, as well as the cell membrane and lysosomes. It named secretory for being the pathway by which the cell secretes protein into the cellular environment. These types of organelle, endoplasmic reticulum, Golgi apparatus, vesicles generally secrete different types of protein into the extracellular environment. Extracellular environment, for this reason, these types of pathway is called secretory pathway. There are two different secretory pathways, regulated pathway or regulatory pathway and constitutive pathway. Secretory pathway are two types. Phases of secretory pathway. Some important phases in the secretory pathway, the initial steps of secretion in the transfer of the protein through the endoplasmic reticulum membrane. The secretory pathway provides a route for the cell to handle things and might not be good to have in the cytoplasm. Another phase, the secretory pathway carries protein to the cell surface membrane where they can be released. Last, constitutive secretory pathway found in eukaryotic cells, in which transport vesicles destined for the plasma membrane leave the trans-Golgi network in a steady state. You read only 2 or 3 specific phases in the secretory pathway. Next, we just read only the definition of signal sequence. What is signal sequence? Signal sequence is an N-terminal short 18 to 26 amino acid sequence that acts like a zip code in nascent polypeptide and determines their subcellular targeting. A signal sequence is a protein region with which a protein can be directed to the appropriate cellular compartment within a cell. So is signal sequence. Signal sequence, we know amino acid have two terminal parts, C terminal and N terminal. So signal sequence is called the N terminal part, and the sequences of amino acid is 18 to 26 sequences, and this sequence acts as a zip code in the nascent polypeptide and determines their subcellular targeting. For the subcellular targeting, this type of sequence is generally used for the detection, determination, or target of the subcellular components. You just read definition of signal sequence. What is signal sequence? Define signal sequence. Next, translocation. What is translocation? Translocation is a type of chromosomal abnormality in which a chromosome breaks and a portion of the reattach to a different chromosomes. So translocation, translocation is a type of chromosomal abnormality. Just a chromosomal abnormality due to the chromosome damage or irregular repair of the genetic material, in which a chromosome breaks and a portion of the reach to the different chromosomes. In which chromosome breakdown occur and a portion of the reattached to the different chromosome. After breakdown of chromosome, some chromosomes attached with the near chromosome. This type of process is called translocation. Translocation process. The term translocation is used when the location of a specific chromosome material changes. The translocation in this example between chromosomes 14 and 21, when a baby is born with his types of translocation chromosome, in addition to the normal 14 and 2 normal 21 chromosomes, the baby will be Down syndrome. You read for detail 6th semester, different chromosomal abnormalities or biochemical disease in that generally occurred by different biochemical or genetic abnormalities such as Down syndrome. Down syndrome such specific symptoms: webbed neck, large chest, low set arms, and others is called Down syndrome. In this definition of translocation, Down syndrome, how translocation occur in the two specific chromosome 14 and 21 that shows in a baby. Just an example under Down syndrome. Properties of translocation. Translocation is a type of abnormal change in the structure of a chromosome that occurs when a part of one chromosome breaks off and sticks to another chromosome. Already we talked, after break down the few chromosomes that it attaches with nearly adjacent chromosome. Translocation occur when chromosomes become broken during meiosis and the resulting fragment becomes joined to another chromosome. So the breakdown occur by meiosis cell division. Another some properties: chromosomal translocation refer to exchange of chromosomal segment between chromosomes. A translocation occurs when a piece of one chromosome breaks off and attaches to another chromosome. Those are the properties of translocation. You read only 2 or 3 easy properties for your exam. Transmembrane protein. What is transmembrane protein? Transmembrane protein is a type of integral membrane protein that spans the entirety of the cell membrane. Many transmembrane protein functions of gateway to permits the transport of specific substances across the membrane. They frequently undergo significant conformational changes to move substance to the membrane. So transmembrane protein is a type of integral protein. Type of integral membrane protein that spans the entirely of the cell membrane. Span entirely of the cell membrane. Many transmembrane proteins function as a gateway to permit the transport of the specific substances across the membrane. So you see, transmembrane protein just specifically work as a gateway to permit the transport of specific substances, not all substances. Transmembrane proteins permits only some specific substances to the membrane. You just read definition of transmembrane protein. Next, transmembrane translocation. What is membrane translocation or transmembrane translocation? Membrane translocation is the process by which peptides are transported across membranes bilayer. In this process, peptides are transported across the membrane bilayer. Membrane bilayer, we know cell membrane, plasma membrane have two layers. This is called bilayer. It is generally consisted by lipid bilayer, and sometimes it is consisted by lipid with protein, and it is called the bilayer. Translocation is mediated by a specific receptor which are restricted in their location to distinct cellular membrane. Short amino acid sequences with a protein known as signal peptide or signal sequences can direct its localization, though translocation also occur in the absence of signal sequences. You just read fast what is membrane translocation or transmembrane translocation. Importance of protein translocation and co-translational translocation. What is the importance of protein translocation and what is the importance of co-translational translocation? First, protein translocation, a significant fraction of the protein synthesis in a cell are inserted into or transported across a membrane. The complex and energy coupled translocation machine. Then it must provide a pathway for those precursors to cross the membrane without compromising the membrane permeability barrier. It is the main importance or function of protein translocation. And co-translational translocation. Understanding the process of protein targeting to a specific location with a cell is a functional part of the biology, a cell biology. Synthesis of protein by co-translational translocation, which also determines the resulting protein topology. Protein translational translocation determines the resulting protein topology. Protein topology, just protein structure, function, properties, and details, is a complex process that is often difficult for students to understand. Topology is a complex process in which describe protein structure, protein function, protein denaturation process, and many other processes. Next, protein transport is defective in the following genetic disease. When protein transport are defected, occurs in the different environmental, physical, or chemical parameters, some genetic disease occurred. First, Zellweger syndrome. Zellweger syndrome, a genetic disease. Next, adrenoleukodystrophy, short name ALD. Refsum disease, Parkinson's disease, hypercholesterolemia, atherosclerosis, obesity, and diabetes. Last 4 terms are easy. Parkinson disease. Parkinson disease is a common genetic disease. Hypercholesterolemia. Hyper. When cholesterol level is high in any organism or any peoples, that is generally called hypercholesterolemia. Atherosclerosis. Atherosclerosis, when LDL types of lipid form a plaque in our blood circulatory system and form a plaque, and it inhibits the blood circulation properly. This type of condition is called atherosclerosis. Obesity, when body weight is when overweight as per BMI and height, then this type of condition is called obesity. Last, diabetics. You know, diabetics, when blood sugar level, glucose level high in blood, this type of called diabetics. You read 6th semester detail, and it may some correlated topic in another semester. Diabetics is nothing. Diabetics when sugar level, when any people consume large amount of sugar, large amount of sweet food, such as potato, sweet, different types of sweet, different size, different types of sweet, potatoes, watermelon, and highly sweet food. Then excess sweet food create an abnormal condition in the body. This is called the diabetics. It is generally regulated by 2 hormone, insulin and glucagon. Insulin generally work for the metabolism of glucose and try to remove the higher level of glucose level in the blood. Insulin work a sufficient number of glucose that is need for the different metabolic process in our body. It is utilized, and when excess glucose entered in the blood, it transferred to cell. It is the main function of insulin. And glucagon. Glucagon is another regulatory hormone that utilizes the sufficient or required amount of glucose in cell for different metabolic and catabolic process. When excess glucose enter in the cell, then glucagon transferred the excess glucose level into the blood. When blood glucose level is very high, then diabetics is occurred, and some symptoms occurred in the diabetic patients: urination, highly starvation, and it is called the mother disease of a living system is diabetics. It is a very bad disease in living system or the human. Next, protein targeting. Protein targeting or protein sorting is the biological mechanism by which proteins are transported to their appropriate destinations within or outside the cell. Protein targeting or protein sorting is the biological mechanism in which proteins are transported proteins are transported to their appropriate destination within or outside the cell. In the proteins sorting or targeting, different appropriate proteins are transported in the outside and within the cell, and also the different cellular organisms, cellular organelles. This type of process is called protein targeting or protein sorting. Translocation of protein coupled to translation. Translocation of protein coupled to translation. Translocation of protein is generally coupled after the transcription process and initial process of the translation process. Most secretory and membrane bound proteins are, we know, most of the protein are synthesis in the ribosome. So we called ribosome is the protein synthesis organ, and some proteins, when synthesis of proteins, some organelles are involved in this process. Endoplasmic reticulum, Golgi apparatus, and vesicles. Most secretory and membrane bound proteins are translationally translocated proteins and reside in the endoplasmic reticulum, Golgi, or endosomes, also used to co-translational translocation pathway. This process begins with the protein in which synthesis on the ribosome. When a signal recognition particle is shortly SRP, recognize an N-terminal peptide sequence. So N-terminal peptide sequence is called signal sequences of the nascent protein binding of the SRP, signal recognition particle, temporary pause synthesis while the ribosome protein complex is then transferred to SRP receptor. On the endoplasmic reticulum in eukaryotes and the plasma membrane in prokaryotes, there are nascent protein inserted into the translocon, a membrane-bound protein conducting channel composed of the ACC secretory 60 on translocation complex in eukaryotes and the homologous secretory OIEC complex in prokaryotes. In secretory protein and type 1 transmembrane protein, the signal sequence immediately clipped from the nascent polypeptide. Once it can be translocated into the membrane of the endoplasmic reticulum in eukaryotes and plasma membrane in prokaryotes by signal peptides. I will provide a diagram, translocation of protein coupled to translation. It is missing now in your sheet. This diagram represent the easy way for the description of the process. Next, post-translational translocation. Even though most secretory proteins are co-translationally translocated, some are translated in the cytosol and later transported into the endoplasmic reticulum plasma membrane by post-translational system. In prokaryotes, in this process required certain cofactors such as secretory A and secretory B, and is facilitated by secretory 62 and 66E, secretory 63. To membrane-bound proteins, secretory ACC 62 and ACC 63 are to membrane-bound proteins, and some also proteins are involved in the post-translational translocation process. I also provide you a simple diagram in the process. Endoplasmic reticulum. Endoplasmic reticulums are a type of organelle found that forms
Sir, sir, 63 key.
ACC 63 and 62 are membrane bound protein. 2 are membrane bound protein that are involved in the post translational translocation process.
Pathways.
ACCA and B and ACC 62 and 63, those are the membrane bound protein.
Membrane bounded proteins.
Yes, membrane bound protein that's are regulated and involved in the post translational due term. Last term of your sheet, signal sequences associated with import into and out of mitochondria. How many signal sequences are involved in import and out of different types of protein in the mitochondria? Last we talk endoplasmic reticulum. You know endoplasmic reticulum is a cellular organelle found in eukaryotic cell that forms an interconnected network of flated membrane enclosed sac or tube-like structure known as cisternae. The membrane of the endoplasmic reticulum are continuous with the outer near membrane. The endoplasmic reticulum occurs in most types of eukaryotic cells. But it absent from the red blood cells and spermatozoa. So we know endoplasmic reticulum types are generally found in eukaryotic cell, and sometimes it is absent in red blood cell RBC and spermatozoa, a primordial germ cell that is produced in the sperm by the spermatogenesis process. First endoplasmic reticulum was observed 1945. Keith R. Porter and Ernst F. Fulham using by electron microscopy. And who is the first name coined of endoplasmic reticulum and with the different functions and structural composition. Endoplasmic reticulum are two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. Mosrine or mosrine, two types. Function of endoplasmic reticulum. The folds of protein molecules in sacs called cisternae and transport of synthesis protein in vesicles to the Golgi apparatus. Next, correct folding of newly made protein is made possible by several endoplasmic reticulum. Chaperone protein, you may read chaperone protein in your third semester molecular biology 2 course inside the chapter 4, molecular organization of gene and chromosome. Next, manufacture of lysosomes enzymes with a mannose 6-phosphate marker added in the 6 Golgi network. Finally, integral membrane proteins that stay embedded in the membrane as a vesicle and bind to new membranes. Initial glycosylation as assembly continuous. This is N-linked types of glycosylation process. So you read endoplasmic reticulum, just a definition. Which scientist first coined the name of endoplasmic reticulum? How many types of endoplasmic reticulum? And 2 or 3 easy function of your exam. It may be short note question. Short note question.
দুই তিনটা পড়লেই হবে।
Yes, short note question. Last term, signal sequences associated with input into and out of mitochondria. Just in this area, we do not read detail, just read how many signal sequence. How many signal sequences? Just only the name. Just only the names, how many signal sequences are associated with the input into and out of proteins into the mitochondria.
স্যার এখানে কেমন কোশ্চেন হতে পারে?
Just name, only short question, 1 or 1.5 marks or 2 marks. List the signal sequences that are associated with input into or out of mitochondria. You just read only 5 to 6 signal sequences name. I will give question in your semester final exam. List some signal sequences or write down the signal sequences associated with into and out of mitochondria. Some signal sequences, most mitochondrial proteins are synthesized as cytosolic precursor containing uptake peptide signals. Cytosolic chaperones deliver pre-proteins to channel linked receptor into the mitochondrial membrane. The protein with pre-sequence targeted for the mitochondria is bound by receptor and the general import pore GIP, collectively known as translocase of the outer membrane. TOM, the outer membrane, shortly TAM, at the outer membrane. It is then translocated to the TOM, the outer membrane, as hairpin loop. The pre-protein is transported to the intermembrane space by small TIMS. Small TIMS, which also acts as molecular chaperone to the TIMS 23 or TIMS 22. TIMS 23 and TIMS 22, TIMS, those are the membrane protein. Translocase of the inner membrane. At the inner membrane, within the matrix, the targeting sequence is clipped up by MT, HSP 70. It is another membrane protein. 3 mitochondrial outer membrane receptors are known. How many mitochondrial receptors found in the cell? 3 mitochondrial outer membrane receptors. First, TOM 70. TOM 70, the outer membrane 70, the outer membrane 20, the outer membrane 22. Those are the mitochondrial outer membrane receptor. And functions: binds to internal targeting peptides and acts as a docking points for the cytosolic chaperone. TOM 20 binds pre-sequences, binds both pre-sequences and internal targeting peptides. Last few lines: the outer membrane channel 40 is a cation specific high conductance channel with a molecular weight of 410 kDa, kilodalton molecular weight, and a pore diameter of 21 angstrom. The pre-sequence translocase translocase 23, TIM 23, is localized to the mitochondrial inner membrane and acts as the performing proteins which binds precursor proteins with its N terminal. In this area, different types of membrane protein with their location, how their molecular height and pore size and some functions in this area. So no need detail to read it. You just read How many mitochondrial outer membrane receptors found in cell? How many mitochondrial outer membrane receptors found in cell with their function? You just write your exam khata. If your question 3 mitochondrial outer membrane, TOM70, the full name of TOM70, the outer membrane 70, the outer membrane 20, and the outer membrane 22. You read only 3 outer membrane receptor name with their function. No need detail in this area. Thank you.